Alright, so if you remember from cell membrane stuff, the cell can monitor what goes in and out. It's trying to maintain homeostasis, it's trying to keep its balance where it needs to be. Stuff that happens naturally, without energy. This is called passive transport. We talked about water molecules being able to move into the cell, basically from the high to low concentration to even out the percent of water or balance out the gradient from the inside to the outside of the cell. We talked about larger molecules being able to move through by the use of integral proteins, stuff that couldn't normally fit through the cell membrane, moving in and out, still moving from high to low because it's trying to meet somewhere in the middle and keep the amounts even. However, sometimes you don't want it to even out. Sometimes you have a lot of something, and instead of evening out where it goes down and hits an equilibrium, you want to get even more of it. That means you have to force stuff in, so it's not just from high to low, it's going from low to high. Now that we know what ATP is, adenosine triphosphate, we know the process of cell energy and how it's made from the aerobic, anaerobic respiration, we can actually see what this ATP is used for. And in this case, it's active transport. So real quick, let's determine the difference between passive and active before we even get into anything. Passive transport requires no energy. It's going to happen naturally, whether or not there's any input of electricity, input of food, it's going to naturally even its way out. This is what happened to our egg in our egg experiment, and this is what happens to cells with water movement. It's always going to go from an area of high concentration or high pressure to low concentration or low pressure. It's going to move from high to low across the gradient. In our case, for the body, the gradient is just the cell membrane. The inside and the outside is going to have different amounts of concentrations or pressures. This can also be referred to as going with the gradient. So that's what we already know. With the addition of learning about cell energy, ATP, we can look at the other side. We can look at the active transport. This requires energy. I cannot perform active transport without some type of energy. Some amount of ATP is going to help me do this. It is actually going to go from low to high. It's going from the bottom of the hill up the hill. It requires energy to go all the way to the top. And it's going to go against the gradient. So there's a couple examples I like to use to show you the difference between passive and active. And these are my three favorite. I feel like this is the easiest to understand. So let's start on the left. Passive, as we know, is going to go from high to low. This is like being at the top of the hill. If I push this ball down the hill, it's going to naturally roll till it gets to the end of the hill. This thing is going to go from a high place to a low place, and it didn't need any energy to get down that thing. On the other side, active, we're at the bottom of the hill. This thing is not going to naturally work its way up the hill. It needs the addition of energy. So in order to get from here to here, I really have to strain and I have to push it. In the cell, this would need a little bit of added ATP to juice it up to the top of the hill. Another one I like is a balloon. Over here on passive, a balloon that's filled up maybe halfway. If I let go of the opening, the air is going to rush out. It is going to go from a high pressure area to a low pressure area. Once I let go of the opening, I don't have to do anything. The balloon is automatically going to equalize the pressure of air without additional energy. If I wanted to inflate the balloon in active, I have to add air with some force from the low area to the high area. I'm going to have to add the energy of my breath. I'm going to have to breathe really hard to fill this balloon up. So that's active transport into my balloon. The last one I use is a river or a current. Say the river is flowing this way on passive and this way on active. In my current over here, my boat is going to float with the current. It does not take any extra energy. I don't need oars. I don't need wind. It's going to float downstream passively without energy. In active, if I want to go against this current, it's a little bit more difficult, but I can get some wind power, maybe some oars, maybe even a motor, but it's energy to work my way against the current. In this example, the current is the gradient of the concentration or the pressure. So we're going to keep this as short and sweet as possible. There are 3 types of passive transport I want you to know, and there's 2 big types of active transport I want you to know. There's 2 parts to the second of the active, but we'll get there. The ones on the left, the passive ones, we already know, but we're still going to go over them. We can go through diffusion. Diffusion of water, diffusion of molecules. That's when molecules are going to basically dissipate and move from that high to low gradient to bounce stuff out. This can be of any molecules. We also learned about osmosis. This is actually a type of diffusion. It's still high to low, but it's only water. Osmosis is the fusion. So if somebody says the fusion of water, that is technically correct, but there's a term for diffusion of water because it's so important, because it happens so often. We actually use the word osmosis as the term for diffusion of water. And then last one is facilitated diffusion. Still high to low, but maybe the molecules are too big or they can't get in through the cell membrane. So they're helped by our yellow guy over here, which is an integral protein. The integral protein is facilitating the movement in and out of the cell. It's helping it out. Alright, so those are the ones we already know about passive. Remember, this is all no energy, no ATP. So let's move to the active ones, the new ones, where we need ATP. So here's the 2 I want you to know. Exocytosis and endocytosis. Whenever we're trying to learn a new biology word, it's always great to look at the prefix and the suffix, basically the beginning of the word and the second part of the word. In this case, exo, out of, endo, into. That's going to tell us kind of what's going to happen. This is exocytosis, something is going to exit the cell, and endocytosis, something is going to come in. And I told you there were 2 of the second one. Within endocytosis, there are 2 I want you to know. Remember, this means getting into the cell. Penocytosis and phagocytosis. So we are moving against the current, against the gradient. It means we need the ATP. So exocytosis, when we're pushing it out, it's going to go from that low to high. This is going out of the cell, out of the membrane. Endocytosis, coming in, but still going against our gradient. This is coming in. But we call this cell drinking. I know that sounds weird, but I'm going to show you exactly why. Phagocytosis is cell eating. So let's look at the active ones a little bit more. We know if we were to zoom into our cell membrane and really look at this thing, it looks like this. It's got the heads, the tails, a couple proteins here and there, but it's basically a phospholipid bilayer. It's 2 lines of lipid. So let's look at exocytosis first. Remember, this means out of the cell. Since it's active and it needs ATP, we know there's a low concentration. We know there's a low concentration there and a high concentration there. So if I want to exit and go from low to high, it's going to require a little bit of energy. Since it can't just push stuff out and flow it through the cell membrane, the cell membrane actually does something cool. It will start to form kind of a little bend or a bubble or whatever you want to call it. Say the molecule we want to move is these blue dots. There are a lot more out here. So if we want to move from low to high, it's going to require energy. And this might be because we don't want whatever those blue dots are. We don't want to have an equal balance of something that might be toxic or some kind of poison, so this would be a good example of why we push against the concentration gradient. It's going to collect a bunch of these little blue dots in a fold of the cell membrane. The cell membrane is actually going to start to kind of pinch that fold until, as you can see, it's starting to almost make like a little bubble that includes the blue dots. Over time, it can actually create a whole separated bubble that is no longer in the cell and spit all this stuff out to get rid of it. It has now successfully gotten rid of 5 blue dots by pushing it against the membrane, by pushing it against the gradient. I now have even less. I started out with a low amount, now I have even lower. Now I have an even lower amount of that thing. This is why it requires energy, because it's not going from our normal high to low. We're pushing up the hill. That is exiting. That is exocytosis, exiting the cell. So now let's do endocytosis. Very, very similar thing. However, there are two types: cell drinking and cell eating. In this case, endo means into. We're going to go from low to high still, since it's a active transport mechanism. So there's a lot of whatever this is already in the cell. However, the cell has determined that it needs more. It doesn't want this to go down and this to go up. It wants this to go up, 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 up. It wants more of whatever this is. Maybe it's food, maybe it's some kind of molecule, maybe it's some kind of nutrient. It wants more of it. So it's like, push more in. Use the energy, push more of that stuff in. So let's say we have some kind of important nutrient as these purple dots. There's a lot of them here, and not nearly as many on the outside. But we want more. We don't want this to even out, we want more. The cell will actually do what's called cell drinking or pinocytosis. The easiest way I always remember cell drinking is like Pinot, like a wine. If you're having trouble remembering which is which, I drink wine, I don't eat wine. So pinocytosis is the cell drinking. The way it does this is very, very similar to exo. It's going to start to fold the membrane into the cell. All the while, any little purple guys are going to be right against the cell membrane, and they're slowly going to get sucked up and engulfed into the cell. It's going to start to pinch this thing off a little bit more and basically do the opposite of exo. Eventually folding the cell membrane into the cell to create a little bubble. Once it has successfully created that bubble and is now inside the cell, you can just pop open one side and all those little molecules spill out into the cell. So now I had a high amount inside, I have an even higher amount by using ATP and folding and forcing stuff in through pinocytosis. The other form of endocytosis is almost identical. It's just about what it's grabbing that makes a difference. Pinocytosis will just kind of grab whatever is available. It'll just suck in all fluid. So if you got a couple of these and you're trying to get more, as you suck in the purples, you might get a green or two, you get some blues. You're basically just gathering a mouthful of whatever the fluid is outside. Sometimes the cell does not want to just gulp up what's outside. It wants to be really specific what it grabs. That's where cell eating comes in, or phagocytosis. Kind of creating a little mouth for our cell. This happens a lot with larger things, and I don't just mean larger molecules, I also mean larger organisms. What if I want to engulf a bacteria or engulf some other large thing? Let's say I got this little guy right here. Some random bacteria that I need to engulf. I need to engulf him because of what he's made out of. Maybe I need to break him down and digest him for energy. Say I've engulfed a bunch of them already that day. So it's going to take energy to grab that guy and force it in. I'm not trying to find an equilibrium or some kind of homeostasis of amount of food. This is like you saying, oh, I don't need food because there's not a lot of food on the table. I guess I'll give up some of my food to the table. No, you want as much food as possible. You want constant influx of something. So in this case, this guy is going to do it. Do the exact same mechanism. Cell membrane starts to fold in. This guy is going to get grabbed up. Cell membrane folds and starts to pinch it off. And before you know it, I have engulfed another little dude, pushing it into a higher concentration of little dudes. Which means you needed ATP to do that. Phagocytosis, eating for the cell. So both of those used the cell membrane to accomplish this. They folded it in and kind of just enveloped it and looped it into the cell, or looped it and pushed it out of the cell. However, there are these things. However, even in passive transport, we used proteins to help us. Active transport actually has proteins helping it also. The one I want to talk to you about is called a protein pump. So a protein pump is going to function a little bit differently than just an integral protein, so I'm just going to make a different color so you know what I'm talking about. It's still a protein. So So a protein pump works a little bit differently than just a regular integral protein. It's going to function just the same way that a pump functions on your bicycle. You have a high amount of pressure of air in the bike tire. Say it's at 20 psi. Say your bike tire needs 30 psi. I'm going against the pressure gradient. If I were to just take the stem or the little thing where you put air into your tire, the air is going to rush out just like our balloon did. I want to pump more air in, so I need a bike pump. The cell uses a protein pump. And the way this works is just like our other mechanisms. And the way this works is just like our other active transports. I have a high concentration of red dots over here. I got 5 of them. I have a low concentration of red dots over here. I only got 1. Say that red dot is really important. Maybe it's something like sodium or potassium. Electrolytes we want a lot of. We don't necessarily want to have a balance of sodium or a balance of potassium. We may need more. We may need more constantly for muscle contraction. So the protein pump is going to grab our little red guy, and then he is going to force him, just like our bike pump, into the rest of the cell. It's going to kind of flip the part that's open and the part that's closed. It's going to kind of flip the part that's open and the part that's closed, and it's going to kind of flip the part that's open and the part that's closed, and push it into the cell, almost acting the same way our cell membrane did in endo and exocytosis in the other active transports. But it's facilitated by a protein. We can't call it facilitated diffusion because we know that that's passive and doesn't use ATP. So when a protein helps actively going from low to high, we say it's called a protein pump. That's our yellow guy right there. Now this can get more complicated, as can any topic in biology, but for now, this is pretty much all I need you to know. Diffusion osmosis facilitate diffusion for passive, exo and endocytosis for active. You can have pino and phagocytosis as the 2 endocytosis parts, and every now and then our active transport will get helped out by our friend, the protein pump.